Today's presentation is What's New in the Simcyp Simulator version 19. Today's speakers are Peter Kilford, Janak Wedagedera, and Mian Zhang. Janak is currently a senior research scientist at Certara's Simcyp division. He earned a PhD from the University of Wales in mathematics and completed a postdoc from the University of Warwick in the UK in systems immunology. Pete is a principal scientist at Certara's Simcyp division, and he holds a bachelor's degree in biochemistry and toxicology concentration with a PhD from the University of Manchester. Mian Zhang is a research scientist at Certara's Simcyp division, and he earned his PhD in DMPK from the China Pharmaceutical University. Pete, Janak, and Mian, welcome to today's event. I'll now turn it over to Pete to begin the presentation.
Thanks, Suzanne. So I'm going to start the presentation off by talking about some of the updates we made in version 19 to the Simcyp compound library. Before I start talking about some of the updates, I would like to just talk through some of the functionality we have for the Simcyp compound library in Simulator. We currently have 42 substrate files for small molecules available in the simulator that can be used. And these can be used in conjunction with 29 inhibitor files and a further 18 metabolites, which can be coupled with the relevant substrates or inhibitor files. Just like to point out that we are actually increasing the amount of biologic compounds that are available for simulations in the simulator, and we currently have 4 biologic compounds that are available to use in version 19. Over the last year, we've been focusing on 2 different projects which have looked at updating and expanding compound files. And in fact, we've updated or expanded 29 compounds over the last year, and they're all available in version 19. And I'll talk through these as we go through the presentation. It's an important job curating the Simcyp compound library. Our simulator is used in regulatory approvals and submissions. And in the last number of years, we've had over 48 regulatory approvals that have used the Simcyp simulator to provide label information on specific clinical drug-drug interaction potential for some compounds, and also to look at clinical dose adjustments for other compounds. So it's an important job to keep the compound files updated and to regularly review all of our historical compound files that are available in the simulator. And this is to ensure that we have the most up-to-date information incorporated into our files. And we achieve this by conducting extensive literature reviews on and evaluating the data that is available. And we also work in collaboration with our consortium members to obtain additional information for some specific compounds. All of our compounds are fully verified to support the regulatory submissions, and I'll talk a little bit towards the end of the presentation around the compound summary documents that are available. So at version 19, we have reviewed and expanded 25 of the compound files that were already available in the simulator, and this has been looking at expanding them to include additional features, as well as reviewing and updating some of the key parameters like distribution and clearance of some of the compounds. We've also added 4 new compound files, and I'll talk through some of these later in the presentation. Simcyp is used extensively by our consortium members and by other members that use the simulator. And in the period from September 2018 to September 2019, there are around 90 publications using the Simcyp simulator in the literature. A number of these are focused specifically on compound models, looking at developing new compound models for specific populations, or for looking at compounds like atorvastatin where specific acid-lactone conversion has been evaluated. And a number of these come from Simcyp themselves as well, where we've published on some of the specific models that we have been working on. For version 19, we've focused on some specific areas for looking at evaluating and updating our compounds. The first of these was looking at a new compound files to support complex interactions, specifically with CYP3A4 enzyme. And the other area for new compounds was looking at food effects and being able to model food effects using mechanistic absorption models. The second area that we've focused on with expanding our compound files has been to look at the ability for it to incorporate complex formulation models using our advanced dissolution, absorption and metabolism, or ADA model, to be able to model both formulation and food effects. We've also looked at expanding a number of our compound files to incorporate renal, OAT, and MATE transport parameters using our the Simcyp mechanistic kidney model. And a number of our compounds have incorporated additional CYP and UGT interaction parameters, where these are now available too. And furthermore, we've expanded the features of a number of other existing compound files and revised them all to incorporate updated distribution and clearance parameters where required. Just wanted to summarize here the compounds that we've looked at as part of the updates of version 19. The first one is coloured sort of the green colour here. These are compound files we've looked at expanding, where additional information has been made available in the literature. We also have a number of compounds where we've revised the files based on distribution and clearance parameters. And the final one, on the blue column on the right, we have the 4 compounds that have been added as new files in version 19. I'm just going to talk through some key examples from each of these areas to give you an idea of some of the updates that have gone into the simulator. The first of these is looking at complex interactions, and I just summarised some of the interactions we've been looking at on this slide here. Specifically, the interactions have been with CYP3A4, where we have drugs that are co-administered, like aprepitant, crizotinib, and rifabutin. Each of these drugs has a complex interaction of their own with CYP3A4. For example, aprepitant and crizotinib are metabolized by CYP3A4, but they'll also be able to induce the expression of CYP3A4 to increase the amount of the enzyme that is present in the liver, but they're able to inhibit their own metabolism by the enzyme, both in a competitive manner and also a mechanism-based inhibition manner, where we see the enzyme levels that are irreversibly degraded, and we have to the liver then have to produce new copies of the CYP3A4 enzyme. And this is coupled with other drugs which are co-administered, like rifabutin, which is also metabolized by CYP3A4, but it can also itself induce CYP3A4. So it's a very challenging situation to be able to model complex interactions like this. And as part of the version 19 project, we've looked at developing new files where we're able to capture all of these interaction parameters. And this has led to 3 new files being added to version 19. We have aprepitant, which has been developed as a CYP3A4 substrate, a competitive inhibitor and a mechanism-based inhibitor, and also an inducer of CYP3A4. It also has interaction parameters incorporated for CYP2C9. This has been added as a research file in the simulator, so it's not available directly in the simulator, but you are able to access this on the members area of the Satara website, along with a number of other research files which have been added both by Simcyp ourselves and also by the consortium members for access. Crizotinib has also been developed and added as a compound in the Simcyp library for version 19. And this, as with aprepitant, has been developed as a CYP3A4 substrate, a competitive and mechanism-based inhibitor, and an inducer. We've also incorporated parameters for the PGP, for being a substrate of PGP for transport into the cells. The final file that's been added on as part of the complex interactions is rifabutin, which has been developed as a CYP3A substrate and also an inducer of CYP3A. And again, as with crizotinib, this is available in the full simulator as a library file. One of the other files that we've added in version 19 is for amiodarone and its metabolite monodesethyl amiodarone. And this file has been specifically developed to be able to predict and simulate the effects we see after amiodarone is dosed in the presence or absence of food, where we see a positive food effect of an increase in the systemic exposure when amiodarone is dosed when we in the presence of food. And the graphs at the bottom here show some of the simulations that have been conducted with overlaid clinical data as the open circles. The solid line at the bottom shows the plasma concentration that is observed after amiodarone is dosed in the fasted state. And the slightly lighter coloured line, the higher absorption here is shown from after amiodarone is dosed in the fed state. It was important to include the metabolite for amiodarone, the monodesethyl amiodarone, as this also has a complex interaction where the metabolite is also able to inhibit some CYP enzymes which are involved in the metabolism of amiodarone and other drugs which are also co-administered with amiodarone. And information will be prepared for this in the compound summary file, which will be available. This has been added as a research file, so as talked about in the previous slide, this will be available on the members area on the Certara website if it's of interest to be used. Moving on now, I'm going to talk a little bit about some of the compound files that we've updated to look at more mechanistic absorption models. The first of these is one of the key inhibitor files that we have for Itraconazole, which is used for CYP3A4 clinical drug-drug interaction studies. The original first-order file that is available in the simulator is still available in version 19 as a default file. And this file was originally designed to simulate multiple dose studies of Itraconazole, and it had very good prediction accuracy. The graph at the bottom here shows the prediction accuracy of clinical studies with the observed AUC ratio shown on the x-axis and the predicted AUC ratio on the y-axis. And it so shows that all of the simulations here fall within a twofold prediction accuracy using the first order model. In version 19, we've expanded the file to allow the user to select the ADAM model to account for dosing Itraconazole as a capsule formulation. And this can be switched on to be able to be used with other complex interactions when we have with midazolam and other drugs which are dosed in specific formulations. Another file that we've looked to expand for version 19, we're looking at the mechanistic absorption, was Ritonavir. And this has been specifically designed to incorporate an ADAM-only file, so that the default file you will see in the simulator in version 19 uses the ADAM absorption properties, and the parameters are all available there that have been fully verified. And this file was designed to describe the exposure of Ritonavir from the commercial amorphous solid dispersion tablet Norvir, and it can be used in both the fasted and fed conditions. The graphs that I've shown on this slide show how the simulations capture both the single dose exposure of Ritonavir after 100 mg dose of Norvir against observed clinical data. And the bottom graph shows the ADAM model, how the new Ritonavir ADAM model performs under the multiple dose scenario. Again, this was dosed at 100 mg QD, dose of Ritonavir Norvir capsule. So moving on, I'm just going to talk a little bit more now about some of the additional updates that have been made to specific compound files. And these updates have really been made to expand our files as more data becomes available in the literature. And the compound files we're talking about on this slide have been specifically expanded to incorporate additional transporter interaction data. And a good example of this was with rosuvastatin, which was originally developed as a substrate for three transporters, BCRP, NTCP, and OATP1B1 and 3. Over the last year, we've updated the model to also include additional interaction parameters for both the OAT2B1 and MRP4 transporters. And this is to account for transport in both from the gut luma and also from the blood into the hepatocytes. Based on the assessments that we did with rosuvastatin, we were also able to update and expand further a number of other compound files that we have in the simulator, including the rifampicin file with additional interaction parameters have been added. Clarithromycin, we've added additional Ki values for OATP1B1 and 1B3. Cyclosporin, again, we've updated additional Ki values for the file. And the Itraconazole file now has additional interaction parameters added for OATP1B1, 1B3, and 2B1. And this has been done for the parent file and also the metabolite, the hydroxyitraconazole metabolite file. I should also mention that the ketoitraconazole metabolite file has also been added in version 19, but this is available as a research file only on the members area of the Certara website. Final part we'll talk about in terms of updates to the simulator, look at the expansion of files to include CYP and UGT interaction parameters. We have 3 files that have had additional interaction parameters for CYP3A4 inhibition for ciprofloxacin to be in line with the FDA guidance for the moderate inhibition that ciprofloxacin can have on the enzyme. Sulfavenazole has been updated to incorporate CYP2C9 inhibition parameters, and paroxetine has been updated to incorporate 2D6 parameters. On the other side of the slide, you see we've updated omeprazole both to include additional mechanism-based inhibition parameters for CYP2C19, but also adding in induction capabilities with CYP1A2. The other file on here is probenecid, which we've added in some UGT2B15 inhibition parameters to fully capture the clinical DDIs that are observed with this compound. Just to mention that all the compounds I've talked about here and the other compounds that are available in the simulator all have full compound summary documents that are prepared and made available on the members area of the Certara website. These are detailed PDF documents which incorporate all the information about the compound files and all of the full verification that's being conducted for each compound. And they're available for you to use and incorporate into regulatory submissions. Just in summary, over the last year for version 19, we've conducted a dedicated review of the Simcyp compound library to support the release of the version 19 of the software. And this has been evaluated with updates and additions to 25 compounds, and a further 4 new compounds have been added and made available for use. The updates have been made to support the use of the simulator for regulatory submissions, and we've really focused over the last year on reviewing and expanding models to incorporate additional interaction and transporter parameters, and to extend the compound files to incorporate mechanistic absorption models using the ADAM model. With that, I'd just like to acknowledge the number of people that have worked on both of the compound teams over the last year, and to now pass on to Mian, who will take us through the next part of this webinar.
Thanks, Pete. Hello, everyone. So in this part of the presentation, I'm going to talk you through some of the new features in Simcyp version 19 with regard to parent-to-metabolite inter-conversion. So firstly, in this slide, we're looking at an overview of the new features introduced by this project in Simcyp version 19. So as you can see with this bullet point, a primary extension introduced by this project is to allow metabolite back-conversion facility in multiple tissues, including kidney, liver, circulation, and gut enterocytes. In addition, considering the significance of flavin-containing monooxygenases in drug metabolism and also parent-to-metabolite inter-conversion, we've also included IVIVE pathways for FMOs into the simulator, together with an absolute abundance-based scaling framework. And last but not the least, we've also implemented a few additional features and enhancements. So basically, we've expanded structure interaction facility and metabolite formation for generic cytosolic enzymes, and also included a common pathway for metabolite formation. And we're going to touch on these new features in the last part of the presentation. So we start with the back conversion facility by looking at some of the examples for reversible biotransformation and also the most common enzymes involved in metabolite back conversion. So as you can see with this slide, so based on our review on those reversible biotransformations, CYP450s, cytosolic enzymes, esterase, and plasma esterases are some of the enzymes widely involved in metabolite back conversion. For instance, CYP450s are involved in the back conversion of tamoxifen and oxide to tamoxifen, cytosolic enzymes in the reduction of 5-hydroxyroficoxib to roficoxib, esterase activity in both plasma and tissues in the hydrolysis of gefibrazole glucuronide conjugate. We've also seen complex cases such as the interconversion between the statin, between a lactone form and hydroxy acid form of statins, where the hydrolysis of the lactone form is mediated by chemical reactions as well as esterase activities, while the lactonization of hydroxy acid has got two different mechanisms involving a coenzyme dependent pathway and a glucuronide intermediate dependent pathway, respectively. So in order to be able to account for these reversible biotransformations within the Simcyp simulator, as I mentioned before, a primary task of this project is to make the metabolite back conversion available in all the tissues where these enzymes are expressed. So here we're looking at what we currently have in Simcyp version 18. So, and what is new in version 19. So basically, as you can see on the left-hand side, at the moment, a substrate within the Simcyp simulator can be metabolized into two primary metabolites, and these two primary metabolites can be further metabolized to form the same secondary metabolite. At the moment, all the conversions, except in the gut lumen, are irreversible, which means that the formed metabolite cannot be back converted to its corresponding parent. So as is indicated with these red arrows in Simcyp version 19, we're going to allow the first primary metabolite of the substrate to be back converted to the substrate. So with this new feature, the user will be able to account for and model reversible biotransformations within the Simcyp simulator. So basically, in terms of metabolite formation, this is something that is currently available within the simulator. So as you can see with these blue lines, with these blue arrows, metabolite formation can happen in multiple locations within the body and within the model. So basically, it can happen in the circulation, kidney, liver, gut enterocyte, and gut lumen. So as is indicated with these red arrows in Simcyp version 19, we're going to, the back conversion option will become available in these tissues so that the users can model reversible biotransformation in all these relevant tissues. Back conversion can be mediated by cytosolic enzymes and esterases in both plasma and tissues. And this will be available in the Simcyp simulator in version 19, and it is considering the relevance of these enzymes in those clinically observed back conversion reactions. We're going to allow multiple enzyme families contributing to the formation of the substrate simultaneously, and this is reflecting the fact that back conversions tend to have complex mechanisms which might involve enzymes from different classes at the same time. And finally, obviously, back conversion can be affected by structural interactions so that the users can evaluate the impact of DDIs on metabolite back conversion. So here in this slide, we're just looking at how metabolite back conversion is handled with the Simcyp models, and we're looking at the simplest scenario as an example where metabolite back conversion happens in the liver with both substrate and primary metabolite modeled with the minimal PBPK model with Simcyp. So with this example, the substrate is given orally and is completely absorbed. CYP1A2 metabolizes the substrate into the primary metabolite in the liver, accounting for 70% of the systemic clearance of the substrate. Renal excretory clearance accounted for the 30% of the systemic clearance of the substrate. On the primary metabolite side, at the beginning, it was only exclusively excreted into the urine. And as you can see with the black lines on the right-hand side, these are the simulations. These are the simulated concentration time profiles for substrate and metabolite without any back conversion happening. So in version 19, you will have this option to introduce a back conversion pathway for the primary metabolite in the liver. And when you have included such a pathway, the rate of metabolite back conversion will be calculated, and that will be fed back into the differential equation describing the liver concentration for the substrate. And with this particular example, after we have incorporated the back conversion pathway, we've got increased exposure to the substrate while decreased exposure to the primary metabolite, as is shown with the blue lines on the right-hand side. Obviously, this is just a theoretical example showing you how the model works. In reality, the impact of metabolite back conversion is obviously dependent on multiple factors, such as the fraction of the substrate metabolism leading to metabolite formation, and also how strong the back conversion pathway is compared with the other elimination pathways for the metabolite. So that was looking at the simplest scenario for interconversion in the liver. Obviously, in Simcyp version 19, when it comes to interconversion in the liver, the user can use either minimal PBPK, full PBPK with perfusion limits to the liver, or permeability limits to the liver to model interconversion. And the point of this slide is that, as is the case with metabolite formation, metabolite back-conversion can always be handled even if different models are selected for substrate and primary metabolites. Obviously, apart from the back-conversion in the liver, we do need to consider back-conversions in the other tissues, including kidney, circulation, and intestinal and enterocytes. So basically, we follow the same principle as metabolite back-conversion in the liver. Wherever possible, when we implemented back-conversion in the other tissues, so as you can see with this fairly complex network, the back-conversion option will be always available for the liver, kidneys, and circulation, with all the possible combinations between substrate and the metabolite models. The only exception is the intestine, where you have to use the mechanistic absorption model, namely the atom model, to describe your primary metabolite and substrate in order to handle the interconversion happening at the enterocyte level. So moving forward, another expansion introduced by this project is the flavin-containing monooxygenases pathways. So here we're looking at a little bit on the background. So basically, FMOs are a major enzyme system mediating the oxygenation of soft nucleophilic heteroatoms within drug molecules, generating oxide metabolites, as you can see with the example of tamoxifen and cimetidine. In recent years, there have been publications looking at absolute abundance of FMOs and also clearance IVIVE for FMO substrates. And all these publications these years have formed the basis for us to incorporate such pathways into the simulator. So in version 19, we're going to have this new FMO tab in the elimination screen where the user can specify enzyme kinetics for FMO 1, 3, 5, and a user customized FMO. We've got multiple options for clearance IVIVE to accommodate in vitro measurements from either recombinant systems or hemolymph microsomes. Metabolite formation and competitive inhibition facilities will be provided. And also, you will have the option to define ontogeny functions in pediatric and preterm populations to account for the age-dependent expression for some of the FMO isoforms. Importantly, an essential component for an IVIVE is the absolute abundance for these FMOs, and this is derived based on meta-analysis of the literature data. This is looking at a few details about the meta-analysis. So for this exercise, for this meta-analysis, we've collected 8 studies with more than 1,000 measurements, applying the predefined exclusion criteria. We've excluded 67% of the measurements because they are samples from pediatric population, 3% due to non-Caucasian samples, and 1% due to poor samples or analytical issues. So the final data set consists of 6 studies with more than 300 measurements, and these are the measurements that went into the final analysis. And here we're looking at the results in the table on the bottom. So basically, in version 19, we've got absolute abundance values for FMO 1, 3, and 5 for the liver and kidney. So as you can see with this table, FM1 has got a 0 absolute abundance in the liver, and this is because FMO1 is a fetal isoform, so its expression in adult liver is basically minimal. For FMO3 and 5, they've got much higher expression levels, basically comparable to some of the CYP enzymes within the hemolymph microsome. For FMO3 and 5, and compared with the liver, the kidney has got much lower and more variable expression levels. So finally, we're looking at a few additional features and enhancements. So firstly, we're going to allow in version 19 metabolite formation and a competitive inhibition of cytosolic enzymes. So in version 19, it will be more convenient for the users if they want to model metabolite formation or DDIs mediated by cytosolic enzymes. And secondly, we're going to have this common pathways feature where the user can use to model the collective formation of the same metabolites by enzymes from different families, including CYPs, cytosolic enzymes, esterases, plasma esterases, gut lumen enzymes and the FMOs. And with that, that's everything I have to say about metabolite back conversion. And I'll pass on to my colleague Janak for the parameter estimation update.
Okay. Okay, thank you very much, Mian. And so this is parameter estimation and automated sensitivity analysis capabilities available in Simcyp version 19. In Simcyp version 19, we have implemented quite a number of new features with regards to PE and ASA. As you see here, the first thing is population fitting. So we have implemented a faster and accurate population fitting method, which is called QRPEM. Is also available in Phoenix and NLME. And then we have also implemented the nonparametric population fitting method. With regards to individual fitting, we have introduced a powerful optimization method, which is commonly used in machine learning community. This is now available in Simcyp, which is called LBFGSB. And we have also introduced method to estimate intra-subject variability, which is useful in bioequivalence trials and other situations where subjects' own PK/PD parameters can vary between different periods or occasions. And then the next one is fitting PBPK models to multiple dependent variables or observed variables. For example, in the human simulator, you will be able to fit parent-metabolite and urine data, and with animal simulator, you'll be able to fit PK and PD data simultaneously. And finally, we have improved substantially the automated sensitivity analysis tool, introducing new global sensitivity analysis methods, three methods, Morris, Sobol, and extended Sobol. So I'll briefly go through each of these areas now. So firstly, why fit PBPK models? As you see in this model fitting workflow, so the clinical data inform at various stages in this workflow. So for example, for model verification and refinement before you apply your model in prediction. So for this, it is very important to have robust and powerful fitting algorithms. So to go through briefly about the population fitting method we introduced in Simcyp version 19. The inter-subject variability in PBPK parameters can be caused by differences in demographic variables like age, sex, ethnicity, or by drug-drug interactions, or by random physiological traits as well. So in this context, it is important to estimate both the unbiased population mean values and also the individual estimates at the same time of the PK/PD parameters. So in this context, the PK/PD parameters and population parameters and observation, they all condition each other. So we have to consider these in building up the fitting methods. So for this, we have used quasi-random parametric expectation maximization, or QRPEM algorithm, which was invented by these people you see in the bottom, Bob Leary and Mike Donlevy, who are members of this project team. So this is one of the most advanced population fitting algorithm available, as I already mentioned, available in Phoenix and NLME. So this is fast, accurate, and will be able to give unbiased estimates. In addition, it can handle hierarchical models, and it can estimate multiple effects on variability. For instance, between subject and within subject, and between period variability, and so on. So what you are seeing here is a sample of Simcyp PE input data sheet. So you have subject ID, time, and the value of the dependent variables, and so on. And in addition, you can specify here the period or the occasion to which each of these observations or each of these records belongs to, so which is clearly shown in this plot. So in the new interface of fitting available in Simcyp, so you will be able to enable estimation of within subject variability, and you will be able to specify various distributions for your parameters, and whether you want to estimate the random effect or individual variability, you can select these from the user interface as you see here. And then the second population fitting method we introduced in version 19 is a non-parametric method based on the NPAG or a non-parametric adaptive grid method, again proposed by Bob Leary and the others, as you see in this publication. So non-parametric methods are quite useful when you have, for example, data coming from populations with subgroups. As you see in this plot, we see a distribution of the clearance of dextromethorphan. So where we have two subpopulations for 2D6 abundance values. So the non-parametric methods shall be able to automatically identify these subgroups within the fitting process. So here we make no assumption with regards to the true distribution of the model parameter. With the parametric method, you essentially need to make an assumption of the distribution as you see here in the blue curve. So this is a part of the user interface, a new user interface you see in Simcyp version 19. So you can select the maximum likelihood as the objective function and the minimization method as the non-parametric. So this is a typical output of a non-parametric estimation fitting exercise we have performed for data coming from two different subgroups. So we have 7 subjects who are CYP2D6 poor metabolizers, and it also shows your initial values and the distribution, initial guess you selected for each of the parameters, and then this shows a reasonable fit with the observed data and the prediction. In addition, it also plots the probability plot for each of the parameters estimated along with the associated values in this table. With regards to improvements we made in individual fitting, so in Simcyp version 18, we had 4 different individual fitting methods as shown in the table here. So in the last row you see the new method BFGS. So this is quite useful in scenarios where we impose constraints or bounds for our parameters. So with BFGS is capable of automatically handling these bounds within the fitting process. So this is a part of the new user interface you see in individual fitting. So you select the objective function as the weighted least squares and the minimization method as a BFGS. And then it has its own termination criteria which user can control by selecting these tolerance values. And this is a output of a typical fitting exercise where it has been able to produce a good fitting with the data we have considered in this exercise. Okay, so the next big item we implemented is the automated sensitivity analysis. So why we perform sensitivity analysis? As you see in this this diagram from EMA guideline in 2018, so sensitivity analysis plays a major role in each stage in this model building process. In the early drug in vitro with early in vitro admit data, and then for those models, it is strongly recommended to perform sensitivity analysis. And then final drug model, it is again recommended to perform sensitivity analysis. So this shows the importance of performing sensitivity analysis. So in version 18 of Simcyp, we had only the local sensitivity analysis tool. So we have introduced global sensitivity analysis tool. So what is the difference between the two? With local sensitivity analysis, as you see in this top figure, the local sensitivity measures only how a quantity of interest or parameter varies locally around the selected parameter space. So because of that, it provides only a limited view of the model sensitivity, because the result can vary within location in the parameter space. So in contrast, global sensitivity analysis, or GSA, determines the overall impact of the selection of input parameters. For example, FU, FA, QH on a specific output, for example, AUC. So GSA method is essentially looking at the variance of the output function, for example, AUC, with respect to the variance in the input parameters. So we have implemented mainly two different methods. First one is gradient-based method, Morris, and the second is variance-based method, Sobol, and extended Sobol. Now here, extended Sobol method shall be able to handle interaction between the parameters. So overall, GSA can determine mainly influential model parameters and their relative impact, and it is useful in identifying influential elements of the model, and also can determine the driving mechanisms for physiological or biological responses. So this is a screenshot of the GSA sensitivity analysis tool interface. You will see in Simcyp version 19. So users can easily and quickly select the parameters for sensitivity analysis just by right-clicking the parameter box on the interface, and then if the number of parameters is more than 2 or more, the GSA tool shall be presented, and then the users can select the distribution for each parameter, lower bounds and upper bounds, and so on. And then finally, with a click of a button, GSA tool can be invoked. So this is an output of from Simcyp version 19 of GSA global sensitivity analysis. So it outputs various sensitivity indices. For example, it shows the first order or main index, which shows the impact of parameter alone. And in addition, it shows a total index for the same parameter, which shows in addition to the impact of that parameter plus the interaction between all the other parameters you selected for the analysis. Now here we show two different plots, this radar plot and the bar plot. So with these, users will be able to quickly identify which parameters are more influential than others in the output. So as I already mentioned, this GSA module can handle the parameter correlations. If you select the extended Sobol method, so there you need to input the correlation metrics. So the rest will be looked after by the GSA module. So in summary, we have implemented a state-of-the-art global sensitivity tool, and then a reliable and accurate unbiased QRPEM method for population fitting, and non-parametric algorithm for population fitting, and a built-in mechanism in the QRPEM to estimate the intocation or within subject variability, and a powerful algorithm to estimate the parameters with individual fitting, and then also capability of fitting PBPK models to multiple dependent or observed variables. And this is the team of our project. And thank you very much for your attention. And we are happy to take any questions from now on.
All right. Thank you so much, Janak, Mian, and Pete. We'd now like to open up the Q&A box to our audience members. Just enter your questions in, and we will try to answer as many as we can. So it looks like we have our first question, and it's a question for Pete. Do you have plans to add the option to use mechanistic atom models for other compounds in the Simcyp library?
Thanks, Suzanne. That's a good question. And our main focus for the next version, for version 20, and the projects we're working on next year are looking at addition of new compounds. Although we are always looking at options to expand the compounds that we have in the simulator and especially on the absorption line. So certainly we're working with the consortium to look at other models that might be of interest to update with adding atom or other parameters into.
Thanks, Pete. Mian, someone wants to know, do you consider the impact of patient genotype on the absolute abundance of FMO3 in version 19?
Thanks, Suzanne. So basically, we're aware that FMO3 is polymorphic, but at the moment in version 19, we're not differentiating the absolute abundance for FMOs between different genotypes. And this is mainly because that we were restricted by the data we've collected. So basically, most of the studies that report absolute abundance for FMO3 didn't really genotype the donors of the liver samples. So at the moment, no, it's not considered. But having said that, we do have the framework to allow the users to enter genotype information in terms of the frequency of different genotypes and the relative abundance of different genotypes, so that they can enter the information if they have the data, and that can be evaluated within the simulator.
Yeah, and it looks like there's another question for you. Someone wants to know, what are the cytosolic enzymes that have been included in the simulator?
So what are the what enzymes?
Cytosolic enzymes.
So we only provide generic cytosolic enzymes, so we don't really specify which enzyme we have incorporated, which are residing in the cytosol. So these are generic cytosolic enzymes, as it is at the moment in Simcyp version 18.
Janak, we have a question for you.
Mm-hmm.
What type of parameter distributions in QRPEM fitting can be selected?
Okay, so by default, users have the, it's made available the log normal distribution for all the parameters. But if the users think they want to assign a normal distribution, that can be, that is available. So log normal and normal distributions are available for parameters to select in the QRPEM. Yeah.
Pete, someone wants to know, they comment, you mentioned the compound summaries are provided for all compounds. Which version of the Simcyp Simulator is used for summaries, and how can we access them?
Thanks, Suzanne. Yes, so the compound summaries will be updated in the version 19 after release, and all summaries will be made available on the members area on the Certara website, so they can be accessed there if they are required, or you can get in contact with one of the team, and we'd be happy to provide more information.
Thanks, Pete. Looks like we have one last question, and it's for Mian. Do you consider enzyme polymorphisms, or do you consider that when you're looking at back conversion?
So basically, for polymorphic, so the answer is yes. So for polymorphic enzymes such as CYP2D6 or 2C19, the impact of polymorphism is is already part of the IVIVE scaling. So it doesn't matter whether that polymorphic enzymes is mediating metabolite formation or metabolite back conversion. The impact of polymorphism is automatically considered because it's part of the IVIVE scaling.
Oh, looks like we got one more question. I think this is for Janak. If you use the non-parametric distribution for parameter estimation, say for a SIP polymorph, do you get two estimated clearance values for each polymorphic variant?
Yes. So let me go back to the question. I can't see it here. Yeah, the answer is yes. I can't see the question. If you use,
Do you want me to give you the ball, Mian?
No, actually, the estimated with non-parametric, the final estimate will be a distribution for each of the parameters. Okay, so it will show the plot of the distribution, and because it's not point estimate you get with non-parametric. The users will be able to select the mode of that distribution if you want to run a simulation. Yeah.
All right. Great. Thank you so much, guys, and thank you so much to our audience for asking those excellent questions. Before we conclude, we have a few short announcements. The next webinar in the series will take place on January 29th when Dr. David Turner will present updates to the Simcyp oral absorption model. On February 13th, Roman Casiano will present the Rare Disease Drug Dilemma: Great Value, High Price, Limited Access. Finally, on March 12th, Dr. Sebastian Pollack will present Virtual Bioequivalence in Dermal Drug Development using in silico modeling and simulation. You can register for all webinars in the series by visiting certara.com/webinars. On behalf of Certara, we would like to thank you for attending today's presentation. This concludes the webinar. Goodbye and have a great day.